Thanks for taking the time to come today. I really appreciate it. So I'm going to talk about blackleg, clubroot, and sclerotinia. And what I'm going to cover today is just a first look at a survey of diseases in canola that's been going on for a few years, and then individually cover those 3 diseases. And within each disease, I'll talk a little bit about it, show you some images to make sure we're looking at this, we're looking for the right things, and then touch on management. And I'm kind of going to focus on some management updates here. So the first thing is this disease survey, and this has been going on for 4 years now. It's the end of the season survey, and the whole concept here is to figure out what we've got going on in the fields, look for any changes, look across the different regions, basically keep an eye on these diseases and the pathogens. And so there's 84 fields from 17 counties. In each of the field, they're going through 50 plants per field, and it's visual identification. There's the breakdown by the map. You can see most of them are most of the field surveyed are concentrated in the northern part of the state, pretty heavy in the northeast. So between 2016 and 2019, there's those 84 fields scouted. Aster yellows is something that we always look for, but it's generally very rare. Occasionally it'll cause problems and you'll see it, and then it'll go away for another half a decade or so, and we just didn't really see much of it. Sclerotinia, so SSR is sclerotinia stem rot. So the incidence and the fields with, so the fields with sclerotinia stem rot, it's fairly common that you see it. There's probably more out there than maybe was captured in this survey. I mean, you can't go to the whole part of the field in every field, right? So, and then the incidence is the number of fields within, or number of plants within. So relatively low, but we have been relatively dry as well. The last thing that I think is maybe the most interesting, and the reason I'm sharing this with you, is the amount of blackleg that we've got out there. So fields with blackleg, that middle column here in the blackleg section, you can see that there's a lot of fields. High percent of fields have blackleg, and some of those are really severe. And when you have that 30% or more, we're really talking about a fair bit of yield loss generally. So we're going to start with blackleg, and in some ways blackleg is maybe the most important disease we've got out there, and it's because some things are changing. So it's a fungal pathogen, and it overwinters in residue. So it forms these structures on canola residue. You have these structures formed, and then they release spores, and the cycle starts. And so these are ascospores, and they can blow around. So in general, the lower stem infections and the early infections, the really early ones in the season, are the ones that cause the economic problem. A lot of the stuff that occurs later on the leaves or whatever is visual, but that's not what's causing the yield loss. And the pathogen is highly variable, so lots of races. The temperature, it'll occur at almost any temperature, which we've got plenty of 50 to 85 degree weather here in North Dakota, and it needs water early in the season. So we do need some rain to get it going. All right, so as you're going through the season, this might be one of the first things you see. So this is a cotyledon. From this point, the infection in the cotyledon can go into the stem itself. And so if close look at that, you can see these fungal reproductive structures, and these will release another cycle of spores, but it's really the first one we care about. So once it goes from the cotyledon, or maybe the first couple leaves, if it gets into that stem through the petiole, it'll start to rot out the stem right away. And when you see it really early, it'll girdle it, and it'll start to disintegrate it, and you'll just lose the whole plant. Later infections will cause the lower stem to turn black and split, and again, you'll see those fungal structures on there, and that's where it gets its name. So this is where blackleg comes from. So with blackleg, during that survey, we saw quite a lot of prevalence of blackleg, particularly in the Northeast. So this is the regions, again, that were surveyed. And the incidence within those fields on average was I would say fairly high. So almost 10% of the plants within those fields in the Northeast on average had blackleg. That's a lot of blackleg. The image here in the corner of the screen was taken from Luis Del Rio Mendoza, the canola pathologist. And each one of those plants has some level of blackleg in it. You can see the blackening inside the stem. So there are 3 new extension publications that were just published in the last month or two of 2020. So Blackleg, Sclerotinia, and Clubroot. And so I'm going to reference these just quickly. So they're available. The county agents in the Northeast will have some of these, but you can get any of these through NDSUACOM, and they're available free online. And so I would encourage you to take a look at that. So using this as an example, we were talking about management here. So you can see in the publications, we always have a management section. Often it's a different color or it's isolated in a way that is pretty visual. So first thing, scout. You should scout your fields every year just to see what's going on, and blackleg is one of the things. If you haven't looked for blackleg, I would encourage you to do so. You manage this with multiple tools, but resistance is probably the most important thing. Crop rotation is really important as well. And then avoiding residue, weed management, certified clean seed, they're all important as well. Seed treatments can help a little bit, but it's not often going to translate into management by the end of the season. But fungicides, early applied, are very important. So I'm going to talk about a few of these. So this is an image that I took maybe 10 or 11 years ago in Minot at the North Central REC, and there was an ongoing long rotation study with canola, and Brian Jenks had a hand in this. So did Art Lamy, who was my predecessor from many years ago. And the image here just shows you the difference in crop rotation. So this was on, I think, year 9 of the rotation study. The bottom field was rotated every 4th year, and the top was rotated half that. And you can see just looking at this, there's a lot of difference going on. And the primary culprit here was blackleg. So I guess as a message here, every other year rotation is good, but if we add another year in it, it's a lot better for a lot of diseases. The next thing is resistance genes, and they're very important. And to give you a visual, I've got 2 tanks here. And so we want resistance genes, and breeders and pathologists and the companies incorporating new resistance genes as warfare, like trench warfare or 2 tanks shooting at each other. And frankly, most of the time the pathogen is winning. And so what's happening here with blackleg right now is the pathogen has races. And so when we're talking about races of the pathogen, and it doesn't matter if it's blackleg or any other pathogen, we are specifically talking about the pathogen's ability to overcome a resistance gene. So overcoming that genetic resistance. Similar idea and concept as a pathogen or a weed or something overcoming a mode of action in a herbicide or a fungicide. The same thing happens in genetics. So the pathogen has lots of races overcoming these genes. In 2004, there was only 4 races that were found in North Dakota. And over the next decade, there was 12 races found. And this is probably going to continue because the pathogen is overcoming the genes as quick as we put them out. So on the other side, you have canola, and canola is essentially, battling this pathogen in trench warfare. But it's the discovery of new genes by pathologists and breeders, and then incorporating them into varieties or hybrids that we can actively use. And that's slow. That's a slow process. And so it's really important once we get a new gene or 2 that we use that carefully. We need to protect that. And we need to rotate the genes just like you would herbicide chemistry. All right. Another thing that I would mention is that fungicides can be very useful here, especially when you're challenged with not having as much resistance in a field or the pathogen's changing quick. So fungicides can be an option. And in general, the early application of fungicides is much better. And there's several effective chemicals, but I'm going to share a little bit of data that was done just recently with Luis Del Rio, looking at trying to refine the timing a bit. So he did a field study, and you can see the information here. I think the most important thing is that he had different times where he was inoculating with a pathogen and then different times when he was spraying with the fungicide. And he's using Quadris throughout. He is using a really hot mixture of the pathogen isolates, so a mixture of isolates. Okay, so this is the first thing of the results. And on this graph here, you can see disease incidence on the left side. So the higher the bar, the more disease incidence. And that's green. Also, you can see disease severity in yellow. So the higher the bar, the more disease severity. And this is related to the time of inoculation. So what this is saying is that the earlier the pathogen gets into that plant, so cotyledon or 6-leaf or whatever, the higher the disease in severity that you have. So inoculation events that occur right away at the cotyledon are going to cause more severity throughout the remainder of the season. And as you might expect then, the earlier fungicide application tends to be more effective. So here you have the same kind of thing where you have incidence in green and severity in yellow. The bigger the bar, the more incidence or severity, and it's organized by the timing of the fungicide application. So an application early at the 2-leaf is more effective, at least in terms of severity, than the 6-leaf. But if you're spraying at flowering, which you might do for white mold, there's essentially no effect. It's no different than the non-treated control. So it's good information to see this, to refine some of those. I mean, we've been talking about that for a while, but this is really pretty solid data that shows that earlier application is a lot more effective protecting those plants. Switching gears here to white mold. So white mold obviously is a problem. So this is a pathogen, Sclerotinia sclerotiorum. Yield loss certainly possible. It's tough to manage, and you might want to consider this management an integrated strategy where a lot of tools might all contribute a little bit to helping manage it. There's a lot of different hosts for this. There's no races in this particular pathogen. The same Sclerotinia pathogen that causes white mold on sunflowers causes it on canola, on dry edible beans, soybeans. So I think it's important to refresh everyone's mind how this works. And so I'll take you through this with pictures. So the disease starts when you have wet soil in the spring, and these small black structures called sclerotia will germinate. So this is the overwintering structure of the pathogen, and these are tough. They can last for many years, but when they're in the upper 1 to 2 inches of the soil and you have wet soil, it'll germinate and produce this structure called an apothecia. So this little tiny mushroom about the size of a pencil eraser. So this will kick out ascospores. And ascospores, they disperse pretty widely, but they're kind of the weak part of the system. So they don't actually infect healthy green tissue. They need something to digest to get the process going. And usually that's floral tissue. So this is an image from Luis. And so he's got a little tiny floret under that white fuzzy growth. And you can see he's got plenty of water on that. So that spore is starting to digest that floral tissue, and it's going to grow right into the green tissue and move from there. Here's another example from sunflowers. So the same scenario, you've got a flower on a leaf, and you can see that pathogen now has progressed into the green tissue and it's starting to grow. You can even see the concentric rings of growth. So the idea is if you're going to prevent this, you got to get it before it's into that healthy green tissue. And then, of course, we all know the story, right? You get a big lesion, the plants will shred, they'll reproduce with more sclerotia, and the cycle will repeat. For this to happen in the canopy, you need it cool and wet. And most of the time, it's relatively cool and it's relatively wet. Okay, so managing this thing. So scouting, we talk about a lot in pathology. Scouting is not helpful with white mold. If you already see the disease, there's not a lot of point in spraying. It's one of those things you make the decision based on a weather forecast to spray or not. There's other things that you can do to help mitigate or manage the sclerotinia and the white mold a little bit. We don't really have resistant hybrids, but some hybrids are better than others. Crop rotation can help, but it's not perfect. Weed management is important because the thing can hit all these different broadleaf weeds. Canopy management, I have that in there just as a caveat that when you have a thick, dense canopy where you've got lots of long dew periods, you're more at risk. So it's not necessarily that you want a thin canopy, but it is important to think that if you're thick and dense and have a lot of yield potential, you're more at risk. So fungicides are a tool, and there's multiple effective chemicals out there, and I would encourage all of you to use the Sclerotinia risk map and risk calculator. This was developed by Dr. Del Rio and with the help of the Northern Canola Growers Association. It's really quite good. The risk map is color coded, so red is high risk, green is low risk, but there's a risk calculator that allows you to put in more information about your field, your rotation, your risk, so it can be more customized to your farm. One note about bloom stages. So most of the labels will say 20 to 50% bloom, somewhere in there is the optimal timing. I tend to think that more efficacy in a favorable environment, you want to lean more early to that window, right? So favorable conditions, you probably want to pull the trigger a little sooner in the window. Again, there's more information that you can check out in this publication that was just put out here in late 2020. Okay, switching over to Clubroot. So Clubroot is a soil-borne pathogen. It's unusual. It's Plasmodiophora, so that is more, it's not a fungi, it's not a bacteria, so it acts a little bit different. And as you know, this was first found in Canada almost 20 years ago now. It was found in Cavalier County in North Dakota for the first time in 2013. It's actively spreading. It likes low pH and warm temps. So it's not to say that this can't occur when your soils are high pH, it's just you're much more likely with low pH soil to see yield loss. It'll take you through the life cycle here. And this comes out of Ohio State. So they don't grow a lot of canola in Ohio State, but this thing will also hit a lot of other crops that are related to canola. And so it's important for them. So if you start with me here in this resting spore stage, what happens right now in March, you've got a lot of these things in the soil, these resting spores. They're going to germinate. They'll form these little flagella, so like little tails that swim and fly around. They'll infect the root hairs. And then once they get into the root hairs, they're going to reinfect and they're going to turn those cells into essentially giant cells full of more spores in the root tissue itself. So they start to swell up and they form these clubs, and that's where clubroot gets its name. What's important here is that those swollen roots at the end of the season are full of spores that are easily dispersed in the wind or through any way, really. All right, so if you're looking at your fields and you see an area, it's thin, it's not looking so hot, you might want to think about clubroot. So this is a field with clubroot relatively early in the season. It's obviously got some damage. This is it a little later in the season. Here's another spot, another field. You can see it's maturing, prematurely maturing. If you dig up those roots and carefully dig up those roots and see swollen clubs, it's time to have somebody else take a look at it and make sure that you know it's clubroot. So later in the season here is after we're very close to the end of the season, I guess, you can see sometimes bare spots, and that's another thing where you might see clubroot. You might notice something's not going on quite right. You can still get into those roots. You have to be very careful because they become brittle and they are full of spores. So this particular root here, you can probably crumble it in your hand, and it's just loaded with the pathogen spores. There's just another example of it. Here's roots that were just pulled and broken off. So again, be very careful to dig into to see it. Okay, so giving you a little information of a survey that was just done recently, several of the extension and research pathologists at the northern RECs were out looking for clubroot, trying to figure out how much is actually out there. And so there's some visual survey and there's also some molecular data. The visual survey is what I'm going to share with you. And I'm just going to share this with you from Cavalier County. And I think this is important because sometimes when you get an introduction of a pathogen, you watch it develop, you watch it move, and it's like forecasting for what could happen in the rest of the state. So this is essentially me telling you to be on the lookout, no matter where you're growing canola. So through the survey here, you have the number of fields in gray and the number of positives in yellow. So these were targeted surveys very early on. But after that, we started to survey additional fields, not knowing whether the pathogen was present or not. And you can see that of those fields in Cavalier County, roughly 20% of the fields had clubroot with no knowledge whether it did or not. So it was first found in 2013, and 7 years later, it looks like maybe 20% of those fields are infected. So this thing is definitely moving. All right, management quickly before I wrap it up. So scouting any time in the season to try to find if you've got clubroot is really important. You do want to minimize the spread. If you know you've got it, you just work that field last, but also be very careful about moving equipment from one side of the state to the other. That's how we see a lot of spread of a lot of different things, everything from weeds to soybean cyst nematode. Fungicides are not effective in this particular group of pathogens, but crop rotations are very important, and so is resistance and rotation of that resistance. So remember the tanks and the races, same concept here applies. So NDSU would recommend, and many others, that if possible, to lengthen that rotation from every other year to every third year planting canola. And a clubroot hybrid with different resistant sources should be used every third year. So not just rotating canola in every third year, but then rotating the hybrid resistance at those third year marks. And that's a much more effective strategy to try to mitigate and keep this thing, to manage it, I guess, essentially. There's another extension publication from Clubroot. I'm not going to talk about that. I would offer thanks to the northern canola growers for putting this on, and a lot of the work that we've done in canola in the last couple decades is supported by them. And then Dr. Del Rio and Dr. Venkat Chapara up at the Langdon REC have supplied some of the information here. I'd also mention that we have good resources in the county agents, and so this map is available. You can just Google the county agent A&R county agents in North Dakota, but many of you will already know the county agents, and they have access to a lot of different information.